Hello, my name is Christina Brown, and this is my presentation on the process of secretion. To put it basically, secretion is the passing of an electrical signal from one neuron to the next. Since the signal cannot jump from one neuron to another, it must be converted into a chemical signal that can diffuse across the space between neurons. Before I show you how secretion occurs, I'd like to go over some terms, then we'll put it all together. A presynaptic neuron is the neuron before a synapse that generates a neurotransmitter. A postsynaptic neuron is the receiving end of the synapse. It may receive synapses from hundreds of different presynaptic cells. A synapse is the junction between the pre- and postsynaptic neurons where secretion occurs. The synaptic cleft is a gap between the pre- and postsynaptic neurons. It is about 20 nanometers wide. Neurotransmitters are chemicals that are released by the presynaptic neuron at the axon terminal. They are how a signal passes from one neuron to the next. Calcium ions contribute to the depolarization of the membrane in the presynaptic neuron. Receptors are chemical gated ion channels found on the postsynaptic neuron. Neurotransmitters bind to the receptors, and the receptors are specific for the neurotransmitter. Voltage-gated channels are activated by change in the membrane potential. They allow ions to enter the neuron that ordinarily would not be able to. In the process of secretion, voltage-gated channels allow calcium ions to enter the presynaptic neuron, triggering the release of neurotransmitters. More on this later. Excitatory postsynaptic potential, or EPSP, is depolarization of the postsynaptic neuron. It causes the membrane potential to move towards threshold, thus continuing the signal on its way. Inhibitory postsynaptic potential, or IPSP, is hyperpolarization of the postsynaptic neuron, causing the membrane potential to move away from the threshold, effectively stopping the signal. So how do we get to the process of secretion? Action potential occurs when the dendrite of the neuron receives graded potentials that summate to depolarize the cell to the threshold of -55 millivolts. The action potential then travels down the axon to the axon terminal where secretion begins. Calcium channels are opened in the axon terminal, allowing calcium ions to enter the cell. The calcium ions interact with vesicles in the cell, triggering the release of neurotransmitters into the synaptic cleft. There are several different protein channels that play a key role in secretion. In the presynaptic neuron, there are calcium ion channels that allow calcium ions to enter axon terminal and interact with vesicles. In the postsynaptic neuron, there are sodium channels that allow sodium to enter the postsynaptic neuron and potassium channels that allow potassium to exit the postsynaptic neuron. What is the difference? If the sodium channels open, the cell will depolarize and move the cell to action potential. If the potassium channels open, the cell will hyperpolarize, moving the membrane away from action potential. There are also calcium channels in the postsynaptic neuron that depolarize the membrane, and chloride channels that hyperpolarize the cell due to the negative ions entering. The receptors on the postsynaptic neuron are specific for neurotransmitters and ions. Only a specific neurotransmitter can bind with a specific receptor. This is a chemical synapse. This is the presynaptic neuron. This is the postsynaptic neuron. This area is the axon terminal, and the area in between is the synaptic cleft. These are vesicles inside the axon terminal, and neurotransmitters are inside of the vesicle. These are voltage-gated calcium channels, and these are the receptors. So now let's look at the process of secretion. First, the action potential reaches the axon terminal of the presynaptic neuron, causing the voltage-gated channels to open and allow positively charged ions to enter the cell. The calcium then connects to the proteins in the membranes of the vesicles. The vesicle then fuses with the membrane of the axon terminal and releases neurotransmitters into the synaptic cleft. The neurotransmitters then diffuse towards the synaptic neuron, postsynaptic neuron, and essentially dock with the receptors. This union causes the chemical gated ion channels to open to allow sodium or potassium to diffuse into or out of the neuron. In this example, the positive ions are flowing in. Any extra neurotransmitters are broken down and recycled by enzymes of the synapse. The last thing I want to talk about is what happens once the neurotransmitters bind with the receptors. You get either an excitatory postsynaptic potential, or EPSP, or an inhibitory postsynaptic potential, IPSP. EPSP occurs when positive ions like sodium enter the neuron through the chemical gated channels. The influx of positive ions depolarizes the membrane, making it more positive and bringing it closer to threshold and action potential. IPSP occurs when negative ions like chloride enter the cell through the channels. The increased negativity hyperpolarizes the cell, bringing the membrane away from threshold and making it harder to send a signal. All of this activity through the channels is summated or combined to reach the threshold and trigger an action potential. In the graph here, excitatory synapses are shown in green and inhibitory synapses are shown in red. If the summation of all the graded potentials occurring at the synapses reaches threshold at -55 millivolts, then a new action potential, shown here in yellow, will be initiated. And that is the process of secretion. Thank you for watching.